Welcome to the PubMed Daily Review for March 26 SD. Today we scanned 3,545 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5. Human brain contusions contain pathogenic transmissible species that induce progressive cognitive decline in tau pathology in mice by Gloria Vegliant and Elisa R. Zanier from Laboratory of Traumatic Brain Injury and Neuroprotection, Department of Acute Brain and Cardiovascular Injury, Istituto di Ricerche Farmacologica Mario Negri IRCCS, Milan, Italy. Let's break down its key contributions. Traumatic brain injury, or TBI, is a known risk factor for developing dementia later in life. However, the exact biological reasons for this link have remained elusive. Our prior studies in mice revealed that a significant TBI event could trigger a prion-like spread of tau protein, which we term tau TBI, leading to memory problems. This new research asks if a similar process occurs in humans. To investigate this, the researchers used a mouse model. They took brain tissue samples from human contusions resulting from severe TBI. These samples were then processed and injected into healthy wild-type mice. The study then monitored these mice for the development of tau pathology and assessed their cognitive function using behavioral tests. The results were striking. Mice injected with human contusion homogenates showed significant and progressive cognitive decline. Furthermore, these mice developed widespread tau pathology, indicating that the tau species present in the human samples were indeed capable of seeding and spreading within a mouse brain. This confirmed the transmissible nature of these pathogenic tau conformers from humans to mice. This study provides compelling evidence that human brain contusions following TBI can contain self-propagating tau species. This finding offers a potential biological mechanism that could explain the increased risk of dementia after head injury. Understanding this prion-like tau propagation opens new avenues for developing therapies aimed at preventing or mitigating the long-term cognitive consequences of TBI. Up next, our second paper out of 5. Intraperitoneal co-delivery of claudin 18.2 times 41 BB and EpCAM times CD3 bispecific antibodies via mRNA-LNPs synergistically suppresses gastric cancer peritoneal metastasis through T-cell coactivation by Jin Ziche and Yang Fei from Department of General Surgery, 4th Medical Center, Chinese PLA General Hospital, Beijing, 100048, People's Republic of China. Let's break down its key contributions. Gastric cancer, especially when it spreads to the peritoneum, is a really tough diagnosis with a grim outlook. Traditional treatments struggle to reach these tumors effectively and often cause significant side effects. We need smarter ways to fight this, especially by harnessing the power of the immune system. To tackle this, the researchers developed a novel approach using mRNA-loaded lipid nanoparticles, or LNPs. These LNPs carry the instructions to produce 2 types of bispecific antibodies, one targeting EpCAM and CD3, and another targeting claudin 18.2 and 4-1BB. They tested this system in mice with humanized immune systems to see if it could effectively activate T cells and fight gastric cancer peritoneal metastasis. The results were really impressive. In lab tests, the E3C4 system achieved over 80% cancer cell killing, a huge jump from the 29% seen with just one of the antibodies, and this was statistically significant. In mice, it led to nearly 99% tumor growth inhibition. Importantly, the treatment was well tolerated, with no deaths and only minor, temporary increases in certain inflammatory markers. This study presents a really exciting new avenue for treating gastric cancer that is spread to the peritoneum. By delivering instructions for 2 different immune-boosting antibodies together in a localized way, they've shown it's possible to be much more effective while minimizing side effects. This could pave the way for a much-needed new treatment option for these patients. Up next, our third paper out of 5. Intracellular fluid accumulation underlies brain volume increases in early Alzheimer's disease by Michalis Kasinopoulos and Natalia Weiler-Theodore. Let's break down its key contributions. In the early stages of Alzheimer's disease, we sometimes see an unexpected increase in brain volume, even as amyloid plaques build up. This phenomenon is thought to be related to neuroinflammation, a complex response involving brain immune cells. Interestingly, when treatments remove amyloid, brain volume often decreases, but it's debated whether this reflects resolving inflammation or actual neurodegeneration. This study aims to shed light on these changes by examining how different water compartments in the brain are affected. To investigate these brain volume changes, researchers studied 297 cognitively healthy individuals. They used a specialized MRI technique called intravoxel incoherent motion diffusion MRI. This allowed them to measure 4 key parameters related to water diffusion within different compartments of the brain, like intracellular and extracellular spaces. They then analyzed how these diffusion parameters related to amyloid and tau pathology, as well as markers of neurodegeneration and neuroinflammation. The study found that the diffusion parameters were strongly associated with markers of neuroinflammation and neurodegeneration, but not directly with amyloid pathology itself. Notably, in brain regions where volume increased due to amyloid, they observed a significant negative correlation with the slow diffusion coefficient, a key indicator of intracellular water movement. This suggests that the increased brain volume might be due to greater cellular complexity or glial remodeling, rather than just fluid accumulation. So what does this mean for us? This research highlights that advanced MRI techniques like IVM can reveal crucial information about neuroinflammation and neurodegeneration, which might be more informative than just looking at amyloid levels. The findings suggest that unexpected brain volume increases in early Alzheimer's could be a sign of the brain's complex response, like glial cells changing their structure. Ultimately, this deeper understanding could help us better interpret brain volume changes observed during treatment and pave the way for more targeted therapeutic strategies. Up next, our 4th paper out of 5. Photoacoustic computed tomography monitors cerebrospinal fluid dynamics and glymphatic function by Songwook Choi and Chulhong Kim from Electrical Engineering, Convergence IT Engineering, Medical Science and Engineering, Institute of Artificial Intelligence, and Medical Device Innovation Center, Pohang University of Science and Technology, Pohang, Republic of Korea. Let's break down its key contributions. Cerebrospinal fluid, or CSF, is constantly moving through our brain, acting like a crucial waste removal system. This process is essential for keeping our brain healthy, but becomes less efficient as we age and can be impaired in diseases like Alzheimer's. Unfortunately, visualizing this vital fluid flow in living beings has been a major challenge for researchers. To overcome these imaging limitations, the researchers developed a novel approach using whole-body photoacoustic computed tomography, or PACT. This technique allowed them to track the movement of cerebrospinal fluid in real time within living mice. They injected a safe tracer, indocyanine green, and used PACT to visualize its journey through the spinal canal and into the brain, providing unprecedented dynamic insights. The results were striking. PACT enabled the visualization of CSF moving from the spinal canal into the brain. Importantly, they were able to quantify how efficiently this fluid was cleared. In a mouse model of Alzheimer's disease, they observed that the tracer remained in the brain for several days, indicating significantly reduced waste removal compared to healthy animals. This study establishes PACT as a groundbreaking, non-invasive platform for measuring cerebrospinal fluid and glymphatic function. It opens new avenues for understanding how brain fluid transport changes with age and in various neurological conditions. This innovative imaging approach holds significant promise for future research into the diagnosis and monitoring of neurodegenerative diseases. Up next, our 5th paper out of 5, Disrupting CD22 cis ligand interactions ameliorates type 1 diabetes and graft rejection by expanding regulatory B cells by Wang Long and Melissa Vasquez Hernandez from Department of Pathology, Nihon University School of Dentistry, Chiyoda-ku, Tokyo, Japan. Let's break down its key contributions. Autoimmune diseases and transplant rejection are major clinical challenges, often stemming from a failure of the immune system's natural checkpoints. We know that CD22, a receptor found on B cells, acts as an inhibitory signal. This study explores whether interfering with how CD22 binds to its natural partners could offer a new way to manage these conditions. To investigate this, researchers developed a special antibody called 1C5, designed to specifically block CD22's ability to bind its natural ligands. They tested this antibody in mouse models, including those mimicking type 1 diabetes and graft rejection. The core idea was to see if disrupting this interaction could alter immune cell populations and, consequently, disease outcomes. The results were quite promising. Treatment with the 1C5 antibody led to a significant expansion of regulatory B cells, which are crucial for immune suppression. Importantly, this translated into tangible clinical benefits. The antibody ameliorated type 1 diabetes in a mouse model and significantly prolonged the survival of skin grafts. Notably, this therapeutic effect was achieved without causing the deletion of B cells. This study highlights a promising new therapeutic avenue, targeting the ligand-binding function of CD22. By expanding regulatory B cells, this approach shows potential for treating conditions like type 1 diabetes and preventing graft rejection. While these findings in mice are exciting, further research will be essential to determine if this strategy can be safely and effectively applied to human patients.